Hey everybody, from the TEN Life Science meeting today. My name is Shane. I run the events over at 10 Capital Network. We have three amazing presenters for you today, as well as some awesome panelists from AngelMD and MD Capital. I'm going to go ahead and bring up our host for the evening, Hall Martin. Get him up here. And one of our actually, we have both of our panelists here now. Awesome. Get them up here. In the meantime, as they're making their way up, please don't forget to put any questions you have in the chat box and answer the poll at the end of each pitch so we can gauge your interest there. With that, I want to let Hall Martin take it away.
Great. Thanks, Shane. I'm Hall Martin. I'm the CEO of TEN Capital, and tonight we're doing a special presentation with AngelMD. We have with us Tobin Arthur and Michael Schmanske, and we're putting on a special presentation here with the joint mailing to both groups. And with that, I'd like to go ahead and after that, we're going to have three presenters come up and talk about their solution in the cardiology space. And we'll be launching and talking about our physician advisory program. With that, let me pass it to Tobin Arthur. Tobin, can you tell us a little bit more about yourself and your group and your messaging?
Yeah, thanks for having us, and we're really excited to have this group organized tonight. I'm going to let Michael do most of the talking. AngelMD loves doing pitch events and loves supporting startups, and we love having organizations that are looking for interesting deal flow and are looking to back companies. And we're fortunate to have the largest network of medical startups in the world. We've got a great network of physicians that help to identify and evaluate these companies and then support them post-investment. But our investment operations and all of that related activity is organized and orchestrated through MD Capital, and Mike Schmanske runs that. So I'll let Mike kind of give a little bit more flavor on what it is we do tactically day to day, but appreciate you joining us and having us a part of the evening.
Great. Thank you. Michael?
Yeah, thanks, Tobin. It's great to see you guys. We're very excited about tonight. This is the first time we've done a co-branded event with AngelMD and with Hall, with 10 Capital and Hall. I represent MD Capital, which is kind of a financial intermediary, which is looking to try to bring together as many of these types of groups as possible and help them service. So while 10 Capital really has the investor network and Tobin and MD Capital as the doctor network, we're trying to act as the in-between. So we're really excited to bring more of these events to you as often as possible. And if you have any interests or specific companies or specialties you want us to cover, reach out, let us know. We're very flexible and we'd be happy to do whatever you guys find most interesting. So tonight we have three fantastic cardiology companies. They are investable. They are seeking money. If you want to know more about them, get your questions into the chat during the session. Reach out to us afterwards, and we will do as much as we can to set you guys up. We also have some evaluation and advisory role opportunities we'd like to tell you about. Specifically, if you are a specialist, if you are a clinical specialist in any of the fields impacted by the companies presenting, make sure you reach out if you would like to talk to the company in future as either an advisory board, a key opinion leader, a beta site, or an investor. And remember, all of these companies are specifically seeking investors in their clinical specialty and are willing to create additional advisory role override warrant pools for those clinician investors. They want you to sit down at the table with them once a month, once a quarter, and work with them as an advisory board member. So with that, I'm going to hand it back off to Hall to kick off the companies, and I'm so glad you're all here.
Great. Thanks, Michael. And so our first company is going to be Sean Morris of Amplify. Sean will be joining us here on the stage as well. He'll have 15 minutes to pitch and answer questions. So Sean, thanks for joining us up here. If you go ahead and launch your slides and kick off, we'd like to hear what you have on Amplify now.
All right, let me go to the top here. So appreciate the opportunity as always. Good to see your some familiar faces. Happy holidays, Merry Christmas, everybody on the front end of December. Seems like it's going by really quick. So I'm Sean Morris. I'm the president and CEO of Amplify Vascular. Just a little bit about myself. I've been in medical devices for 25 years. The last 15 have been running startup companies. I've been fortunate enough to have some exits under my belt, which has given me a great opportunity to put my chips on bets that I think are going to be really big and return capital to our investors. About a year ago, I went and took over a company and pulled a really amazing asset out of that. It had already been funded really in an A round all the way through a first in human study conducted by Dr. Adrian Ebner in Paraguay. And I'll get into that in a minute. I want to give a little bit of background about the company. So since then, we've been really focused on taking this technology through the FDA approval process, and then to add on to our already pretty amazing clinical data in a space that is in big need, a space that I'm passionate about, and it's in my sweet spot, which is vascular. Let me make this a little bit bigger so we can see the slides larger. So we are a device, medical device, feeding a big TAM with a large growing market. Again, first in human data, our reg path will be de novo 510(k). We do expect to have our IDE approval Q1 of next year. We have a really great team of solid engineers, lots of folks with LVAD type backgrounds, so great blood pump technology expertise, really good IP, FTO. We have many large med tech companies around the table that we've already introduced technology to, such as Medtronic, Abbott, Terumo, Gore, etc. We're looking to raise $7 million in a Series A. We have 5 of that, so a good chunk of that already under our belts. We have more at the table. What I'm looking for is great clinicians that can help us, not only to bring us further down the point, but I'm looking for smart money too, people that can dig in and really understand the space. I believe we have opportunities for an exit by 2025. So the premise is in our area of focus, which is bypass and vascular access. There's a lot of papers out there. I've just referenced a few that really talk about the starting vein diameter is a great predictor for bypass and AV fistula access outcomes. In other words, how well do they work? How long do they work? And do they even mature to the point where they're usable, specifically, which is a big deal in AV fistula for dialysis access? There are many forms: CABG, PAD, FEMPOP bypass, or when a physician, vascular surgeon creates a fistula for dialysis access. So our initial focus is going to be on AVF, and everything else will make a lot of sense after that. Looking at a big market, $36 billion spent on end-stage renal disease, 10% of the CMS budget, yet only 1% of the patient population. And there are 3 major forms of access: dialysis catheters, which are prone to infection. They're not the ideal candidate, but they're really good for sort of a bridging hemodialysis access strategy. Grafts, upper arm fistulas, and forearm fistulas. Forearm fistulas are by far the best. They have the lowest morbidity and mortality, lowest cost of care. If you can get them to work, they're the best form of access. They can take a needle stick and keep on humming. But they have a high failure rate, and really a lot of that goes down to vein size diameter at the beginning. So patients present with end-stage renal disease, they need access, they go to a vascular surgeon to get vein mapping, they find candidates for good access, many of which are very small. So if I talk to a vascular surgeon, they might say 2.5 millimeters of starting vein diameter is really where I make my decision whether a vein is a good candidate or not. Otherwise, I'll go to the upper arm. When you do that, you've done a disservice to the patient, because now you're eliminating lower arm as a potential access site for their dialysis access. So we focus on using our technology to make these veins larger, and I'll get into that. And really, it's predicated upon KDOQI, or Kidney Dialysis Outcomes Quality Initiatives guidelines, or University of Alabama Birmingham guidelines. The rule of 6 millimeters in diameter, 600 milliliters of flow, or 5 and 500. There's a great paper sponsored by the NIH that looked at starting vein diameter is critical for suitability and maturation success. So on the x-axis here, we have starting vein diameter as a predictor for achieving these KDOQI criteria. So basically, about 3 millimeters in diameter, your predictability of success is very low. As you go out the x-axis, you see larger starting vein diameters, you have more of a success. So our technology essentially takes these candidates down here and moves them to the right. And the way we do this is using a blood pump and extracorporeal, so think of ECMO type blood pump on the right side of the heart. A patient wears this for 24, 48 hours. It siphons blood out of the SVC right atrium juncture, just like a dialysis catheter would, into a pump, and forces it down into a very small stick into the a vein candidate in the lower arm. Over time, this vein dilates, much like what happens when a natural AV fistula is created, a surgical AVF, where you connect an artery to a vein, except for that's pulsatile flow, and it could take 12 or more weeks. And in the forearm, it's less than flip of a coin whether that outcome occurs. So what we do is we can superimpose this with a mechanical solution. Patient wears this again, it's taken off, and then the AV fistula is created with an already large vein, which we know will impact the patient and their outcomes. Conducted a study with Dr. Ebner in Paraguay, Dr. Shenoy is from Washington University, St. Louis. He's been in this space for about 5 decades. And you can see a venogram of a starting vein diameter starting pretty low. And just with treatment, you can see it getting big and robust and actually a long-term segment. The data was very, very intriguing. On the right side, starting vein diameter is baseline 2.3 millimeters. At the end of the treatment, 5.3. After conversion to an AV fistula day 1, it maintained itself and actually got over that 6 millimeter and sustained at 2 weeks and 6 weeks. And these patients are actually getting dialysis even more than a year out from the procedure. Blood flow, same way. Starting blood flow going in after treatment over 700, again surpassing UAB and KDOQI guidelines. Day 1 sustained, week 2, week 6. These are high flow rates. This is making the dialysis centers and the patients really, really happy because they have a good clearance, which is how in the States these patients get paid, or these doctors get paid. From a treatment time, we can look at the histogram here and we can see at 12 and 24 hours, we're already there. We're already pumping. Now, these patients were kept on a pump for longer because we had less data then, but all of our patients achieved these very, very soon, which is important for the clinical study and for the eventual economics of it. And we know that it's really wall shear stress over time is which stimulates a vein and makes it grow to accept more flow. Many opportunities to talk to many of these people, all of which, except for one that I have thus far, sometimes more than one time. And I've only been really in the seat here for about a half a year working on this as the CEO. We believe we can turn access and bypass upside down with this revolutionary technology, which really is using a physiologically and hemodynamically known system that increases vein dilation. So it's going to have a big impact on patients with less failures, and it's going to be less needy, and it's going to take dollars out of the equation as well. From a timeline perspective, here we are looking at Q3 this year, done a lot of the work to prep the IDE with all of our non-clinical and benchtop testing and, the FDA stuff. And then taking our first in human data, we've had 4 meetings with FDA in the past 12 months, and we're ready to submit really in December. So probably after Christmas. We could do it before, but we don't want to make FDA upset with us. They have 30 days to review the IDE and get back to us. And we built a really good relationship with them. We'll start with 10 patients. And after that, we will go into our randomized study where we have a 2-to-1 randomization, 2 treated with Amplify, 1 to standard of care. And we believe the biostatistical data will really root out, really if we increase, if we move the needle 20, 30%, it's pretty groundbreaking in terms of giving these patients a better opportunity, not only increasing the number of patients that can get a fistula, which is important, but also making sure that those fistulas that are going to be used for long-term access are larger caliber vein, because we know that it has a big impact on outcomes and usability. I won't get into this real marketing slide, but we believe we have a robust business. We believe even at an ASP, which we have modeled even higher than this at $19,000, that there's a lot of opportunity for upside and a really, really nice margin for more than likely some other company that will acquire us after we complete our study. If we are going to go commercialize that, then that'll be a very strategic decision. But I believe based on some good inertia that we'll be an acquisition candidate after we complete our study. I couldn't say it any better than Dr. Shenoy is poised to become the most significant discovery in the field of vascular access since AV fistula was first described in 1966. Dr. Shenoy has been involved with lots of companies, but he's never been a part of one from an industry collaboration perspective. We'd be the first. Here's our team. Paul Muller's our Chief Technical Officer. Erdi Di Peralta, again, great experience with blood pump related technology. So testing and clinical, these are implanted LVAD technologies. Ours is more simple than that. Great IP. From a financing perspective, I went over this, but it's really a flat round from our C to A round. And we're going to use the funds just to focus on clinical data and get it through the FDA gauntlet. With that, I'll yield.
You'll yield the remainder of your time to the young man from Pittsburgh.
Exactly.
Thanks, Sean. So 25 years experience. Was that headshot in your bio from the beginning of those 25 years?
It is. Yeah. I had more hair. I have the COVID beard, which my wife likes. So I just but yeah.
Very good. Well, this is fascinating stuff. Now, one of the things that struck me when you first presented this product was trying to understand a little bit more about the safety of inflating these veins. Like you yourself mentioned, it's not, you're not actually blowing them out, right? It's not this pressure that's opening the vein. It's the wall shear, the amount of flow.
That's correct. Yeah, that's correct. That's a good question. So this is a big curiosity for me. And what I like about medical devices, just at a high level, is that it's a mechanical solution to a plumbing problem. And I've developed balloons, stents, catheters in my time. And a lot of times we're either trying to open one or we're trying to close one. With the a lot of the data with when a physician connects an artery to a vein, you're now putting in a lot higher flow into that vein. So that vein's got to accommodate that. And the way that it does that is it dilates, it expands. And there's really a biphasal approach. There's the initial expansion, and then there's conditioning or arterialization of that vein, which occurs by virtue of this. So we're just shoehorning a mechanical solution that is more predictable and faster than the typical just pulsatile blood flow from an artery.
And the body naturally knows to pick up the process once you've increased the flow. It just automatically responds in that way and stabilizes it at that level.
That's right. Yep. And there's been some, long studies a long time ago with high injection of saline and some other things in there to dilate. But we're taking blood and we're putting blood in. So we're basically creating a venous to venous conduit, and we're putting, venous blood into that vein. And over time that stimulates and expands to accommodate that flow. But eventually we will connect the artery to it, or we won't, but the doctor will. Right. And then you'll have that arterial flow going in there. And then the vein itself becomes arterialized. They call it venous arterialization.
And this has been done with other mechanical support before. Your innovation is in the way you're doing this. Tell me a little bit about what differentiates you from existing AVF treatment.
Yeah, so we have tested our pump, and actually the pump itself spins faster in recognition that the vein gets larger. So we want to keep that volume pounding against that vessel wall, because we know that it's against that wall shear stress against the vessel wall that stimulates it to make it dilate to accept the higher flow rate. That's well understood. Every time a bypass is done, that's what happens. Every time limb preservation procedures are big now, where you're using a vein and turning it into an artery for the lower limb to preserve the leg, and this limb flow technology that was actually recently acquired by Inari, which is a great comp for us. Okay. So it's a well understood mechanism.
So every time you answer a question, I want to ask another question. I feel like I want to jump in here. But and by the way, docs and everybody else that's watching tonight, feel free to definitely get your questions into the chat room. I'm happy to have this conversation with Sean myself, but I'd love your input and love your questions to get in here as well. So this has been, AVF has been pursued now for, you said, I believe the first data was in the 60s, 1967 or something, you said?
Yeah, the surgical procedure for this application was described, and it's in the literature since the 60s.
But it's full, like the current means of vein dilation are approved. So we have long-term stability studies. And I guess what I'm trying to get at, let me just jump to the question rather than prefacing it.
Sure.
Are there any concerns that the FDA would require a much longer post-procedural monitoring period in order for you to achieve clinical approval and 510(k) approval? Or do you feel confident that the regulatory phase will be limited, or scrutiny will be limited to the dilation phase of the trial?
That's a really good question. So we have worked with FDA, of course. As I mentioned, we've had 4 meetings, some of which have been, hey, this is our design and our protocol, predicated upon what we know from lots of clinical data, which is suggestive or shows that what a vein looks like and how it's acting 1 week after AV fistula creation. So again, surgical baseline, standard of care is a big predictor of how it's going to function at 6 weeks and then beyond. So 1 week, 2 week, we're looking at a 2-week milestone. So that's our endpoint, 2-week maturation endpoint. And when we compare that to what standard of care, when again, just a simple connection between an artery and a vein, we know how poorly they do. I mean, which is why we exist, right? Because we know that we can do better. When we compare standard of care versus amplified, treated, and then converted, we believe we'll come out as a big winner. And then From an economics perspective, of course, we'll follow these patients out longer, because that's also part of our value proposition, is how functionally mature and how long do these last from a primary or a secondary patency perspective, which is when inevitably clotting will occur, or you'll get what they call stenosis, or a narrowing between the, usually at the anastomotic site where the artery is connected to the vein. Those wear down over time. They're stuck with needles by people who make $17 an hour. So there's going to be a primary, endpoints, secondary endpoints. So we believe that a larger vein across the board, again, data supports this, they do better primary and secondary patency as well.
And to your knowledge, the FDA acknowledges that this is a procedure that should be promoted. Yeah, like they're in favor of the product.
We know, this, it's hard for me to say this and actually believe myself when I say it, but FDA has been very collaborative and I would say excited. One of the reviewers, or the vascular reviewer, is Dr. Bob Lee, who is a vascular surgeon. And so he knows the benefit of this with his background, and they're very supportive. And we believe that, I always fail to mention this, but it is important that we believe we'll have breakthrough designation, which will give us a faster pathway through the FDA process, but also from a reimbursement and access to CMS. So again, the economics, 10% of CMS's massive budget, taxpayer-funded healthcare, goes to 1% of the entire population under the CMS umbrella, because dialysis is so expensive, or end-stage renal disease is such an expensive thing to cover.
Got it. That was actually going to be my next question was, well, in a gross fashion, how do you get paid? How do we get paid? But in a detailed fashion, do you have approval codes, and do you believe that all of this will be supported by the insurance companies?
The latter, yes, I do believe, and it's up to us to prove it clinically and do the blocking and tackling and what we need to do. There isn't any existing codes right now for our technology. It's novel, it's new, so we will have
breakthrough designation will be very important for that.
Absolutely, yep. And that's intimidating to some people, right? Because you always talk about the 1R, which is regulatory. That's tough enough, but reimbursement is another sort of layer. But I look at it, hey, tell me a strong company with a large exit that didn't have to do that. I mean, it's a novel technology that's solving a very, very large unmet need. This is not an angioplasty balloon that has a little bit more atmospheric pressure potential. It's not another mousetrap. It's the juice will be worth the squeeze, but just like anything else, we'll have to do a little bit more work. But doing that is going to create a larger exit.
Talking about patent protection and your IP, like your patents are on for the methodology or on the whole process. Like, how difficult would it be for somebody else to come in with an improved pump and just step in there?
Yeah, so we have a really, the company that we pulled those out of spent a lot of money on IP, and really a lot of money on lots of things. But IP was one of the areas where, hey, I'm happy that they did that, and we have freedom to operate. It's the technology, but also methods, which you can't get method patents around the entire world. It's more like just the United States and a couple of other countries, but we do have both, and they're robust. And we do this. I'm smart enough to realize if we do this the right way, there'll probably be some other competitor or whatever come down the road, but then they'll have to cross over whatever hurdles we create. We'll be the first to market, and they'll have to do a lot of heavy lifting over and above what we do. Otherwise, why would anybody switch to something different? Got it.
Well, you know what? We are running up the time, but I do want to ask two other questions. First is, how do we get paid? How do the investors get paid? Like you mentioned having a comp of another company that was just acquired recently. Like, that's your target, or self? Like, what are you looking to do, and how?
So I truly believe that this is a $500 million plus opportunity, and what's that based on? That's based on the amount of opportunity there is in terms of the market, the opportunity, but also there's some comps. And I mentioned LimFlow because that's a very near-term comp. That's a vascular surgery call point. It is when a doctor is connecting an artery to a vein, or turning a vein into an artery to get perfusion down in the limb. There's a lot of things that are similar. You could also look at Silk Road Medical, which is using a carotid access for carotid stenting, as another great comp, which is a publicly traded company. That's also another vascular surgery call point. And I'm only talking about AV fistulas. When I talk to a vascular surgeon about this, they're like, well, I would like to use this on a saphenous vein for cabbage, or I'd like to use this for bypass. So there's a lot of other upside potential that's not even included in this available market. I joke and I say, if you find me a vascular surgeon that doesn't see value in this, I'll cut one of my fingers off my hand, because I haven't seen it yet. So I'm that confident.
Hands up, Sean. Hands up. Let me see him. All fingers. Very good. Excellent. So one last question for you, and this is really targeted for our audience as well.
Yep.
You are really, really, really interested in having specialists in the field as your investors, right? Can you tell us a little bit why and how you want to interact with them? Like, what is it that you're looking for from the docs, and what do you expect from a doctor investor?
Well, I mean, I look at it very pragmatically. Like I've done this before. I've had my last company. I brought in physician investors for a stroke company that I built, and they were stroke folks, but they were also cardiologists, because there's applications for limb preservation and critical limb threatening ischemia and lots of other applications. So what I find is that, why not invest in something you actually know something about, or you can have an impact with whether it's clinical trial design, or whether it's helping me along the way with some critical steps, or maybe you have some insight from some other companies you've seen, or regulatory considerations for us. And then you might be our customer too down the road. So you can really help have an impact on your own investment throughout the entire value chain. And I would pay you anyways, from an equity perspective. So what I like to do is if I have a physician investor, I like to incentivize them even over and above their investment in equity by creating an advisory pool over that investment to doubly incentivize. Skin in the game is important because you want to be helpful, but it's also I would want to use you anyway. So that's where there's a lot of synergies.
Well, MD Capital, like one of the things we are specifically looking to do is creating management wrappers, where we can essentially take a syndicate, you add the extra layer of warrant override that the company provides, and we use that to compensate the investors with an extra amount. So you invest $10,000, you're getting $11,000 of equity, and that extra equity represents your advisory coupon, your payment for being an advisor to the company. Obviously, we're looking for minimum investments larger than 10,000. I just want to make it an easier number.
Yeah. Well, that's exactly right. That's exactly right. And then my job is to execute on a day-to-day basis, get the good clinical data, which again, for me is the most important thing. Everything else is noise. You drive clinical data. And then when somebody buys us, then that distribution goes back, and there's a little extra push because of the equity that we would provide to our advisors.
Got it. So I want to lay that down again. If you are in a specialty that works with these devices, that is interested in this product from a professional level, make sure you reach out and contact us, because we have extra bonus equity for those specialties, because we want you as key opinion leaders and early adoption drivers. Now, this is going to be true for all of our companies tonight, and this is going to be true for many of the companies that we bring out this way. So make sure you sign up for an ongoing newsletter for the companies that we bring as well. There will be an orthopedics in the spring. We'll do more cardiology companies in the future. Now, with that in mind, I'd like to hand this back to Hall. I believe we have another company coming. Thanks so much, Sean. Is there something else that you'd like to close with us before we leave? Because thanks so much for the presentation. Very exciting stuff.
No, I'll yield to the young lady from Pittsburgh, but I really appreciate it. Doctors are my customers, right? And I love the creation of these devices and finding solutions to use devices, and there's a lot of creativity down there, and people who are treating patients on a day-to-day basis. I always try to do things that are going to help them, but it's this. So I appreciate the opportunity, and I'm here for any other questions. But good luck to everybody, and happy holidays.
Great, thanks, Sean. And with that, we'll have you go back down the backstage, and we're going to have next James Blackledge of Capella. James, if you could join us up here on the stage, love to learn more about your opportunity there as well.
Okay. Thanks, everybody. I appreciate the opportunity to talk to you today about Capella Imaging and what we're doing with our lead candidate, FiberScent. Capella is a small startup based in St. Louis, Missouri. We grew out of the Washington University Medical School. Founders have a great deal of experience in drug development and cardiology. We are all research scientists at heart with experience ranging from discovery to commercialization. And our lead candidate is a nuclear imaging agent we call FibroScent that is used for the assessment of both acute and chronic venous and arterial thrombus with a number of different indications. Our initial indication is for detecting thrombus associated with LVADs and the quiescent regions of the heart. But as you'll see, there's a number of follow-on indications from prosthetic valves to pulmonary embolism, DVT, and even some things associated with cancer. Detecting thrombus is a challenge with many different cardiovascular diseases. Cardiovascular disease is a critical problem in the U.S. 20% of the mortality is based on some form of cardiovascular disease, which has resulted in a growing population of people that need some form of mechanical circulatory support. And these people are placed at an elevated risk for thrombus. And there are many different aspects of this that are difficult to diagnose. For instance, with LVADs, and especially with some of the earlier, the HeartMate 2, HeartMate 2 was just riddled with clotting problems. HeartMate 3 does considerably better. But these are, essentially titanium pumps that are implanted, and you can't image with those devices and look for clots. We can actually image clot within the titanium shell of these pumps. We can detect clot in the quiescent dead regions around the heart. So we're able to effectively diagnose and locate thrombus in a number of different forms of cardiovascular disease. And we think this represents a tool that doctors can use for better patient management and also for managing the associated anticoagulant therapy, because most of these patients that have mechanical circulatory support are going to be placed on some form of anticoagulant. And it's a balancing act between, preventing clotting and the risk of gastrointestinal bleeding and the associated hospitalizations with that. So this is an image that we acquired from an LVAD from a patient that came into the Washington University Medical School. The pump was suspect, and they were scheduled for surgery to replace the pump. We took the pump out and placed it in our mock loop and acquired this image using FibroScent. And you can see in the neck at the bottom of this rotating image, the thick layer of thrombus that is deposited on this device. And, this patient was at risk for having some of this break away and causing a stroke. We think this represents probably the most challenging, the worst case scenario for getting an image. This is a very high shear environment, a very high flow rate, high levels of anticoagulant. In many cases, the clot that is deposited here is old. And, there are a number of agents that have been reasonably effective at imaging acute clot. They're not very good with chronic clot, and we do very well with both. So again, we think this represents the worst case scenario, and we do very well with it. So there's really, again, our initial indication is LVADs, and there's a lot of unmet potential here. There's at least 25,000 patients annually that need some form of mechanical support. The gold standard for heart failure is heart transplant. However, there are only about 3,000 donor hearts available annually, and this number is just not changing. It's been stagnant for quite some time. So the rest of these patients need some sort of solution. Because of the increasing efficacy of LVADs, the allocation strategy for donor hearts has changed in recent years. They used to be essentially bridge to transplant, meaning this would keep you alive until a donor heart became available, which is statistically unlikely. And so there's more of an emphasis on destination therapy, where you're going to live with this device. You're not likely to win the Ironman competition, but you can live a healthy, productive life. And so there's, again, this growing population that needs some form of mechanical support and requires better tools to manage these patients. And we think this is one of the things that's hindering the market right now, is the lack of the effective tools to evaluate and manage the anticoagulation therapy of these patients. And we think FibroScent is a potential answer to that. So looking at just the LVAD and prosthetic valve markets at this point, there's about 190,000 patients each year that are added to this pool. We envision, at least initially, 2 images per year, so that's 380,000 images. And so $1,500 reimbursement per dose, and this represents a market of about $550 million annually. That's a total addressable market. There's a fair amount of, I think the cumulative growth rate of both of these markets is around 7%. But there's also follow-on indications, as I mentioned before: pulmonary embolism, DVT, and peripheral valve or peripheral venous thrombus associated with cancer, which is a very large market. And there are aspects of these, there are good tools in place for a couple of these now, but there are aspects of all of them that are very difficult to diagnose. And we think FibroSent represents a very good way to address those difficult to diagnose problems. What's key is to recognize that once you get your foot in the door there and you establish the utility of this for imaging, you have the potential to become the choice of first pass for diagnosis. If you recognize that in 20% of the cases, I'm potentially going to have to do a follow-on image with FibroSent, or in the case of echocardiography, where often it requires multiple forms of echocardiography to get a good diagnosis, for instance, with prosthetic valves, and you can address this strictly with one image from FibroSent. You have the potential to become the tool of first choice, which represents a very large market. So what I really want to stress here is that FibroSent has been heavily de-risked. This is a cartoon image of FibroSent. It's basically a 4-arm tetramer. Each arm is terminated with a peptide that recognizes fibrin, and another peptide that chelates the radioactive technetium, which is the label. And so most drugs fail in development when they transition from preclinical to clinical, either due to efficacy or tox. And in this case, as a nuclear imaging agent, the amount of material you dose is exquisitely small. And so the risk of toxicology is very, very small. And in fact, the agency recognizes that. They only require a single species of tox to open an IND. We have done the GLP toxicology study and got a completely clean bill of health. The other risk is efficacy, right? I like to say we've cured cancer in mice many times over, but a lot of drugs fail in the transition to humans. In our case, one of our preclinical models is we take pumps out of patients that have come into the medical school for exchange. We implant those in a 70-kilogram calf, which we use as a model for human in terms of blood volume and flow and mass. And we acquire our images from this model system. So these are actual pumps that have human clots have been removed from human patients. So this is as close to clinical data in a preclinical model as you can get. So again, this is highly de-risked in terms of moving forward to approval. So in terms of our strategy to getting to market, our customers are the physicians that treat these patients, and cardiologists that implant the devices and are responsible for maintaining them subsequent to the implantation with all the anticoagulant therapy. So our plan is to get approval, to obtain the appropriate reimbursement codes, and we want to partner with a third-party provider such as Curium, GE Healthcare, Cardinal Healthcare, someone that already has an existing network for distribution and sales and promotion that we would partner with for actually distributing this and getting it to the patients. And I will say a few of these have already reached out to us and expressed interest at this point. It just seems clear that everybody's kind of waiting to see some clinical data. Competition in this space, in the case of VADs, there is nothing. It's a completely unmet medical need. Again, in the case of prosthetic valves, pulmonary emboli, it's the standard of care, and the standard of care works well for the bulk of these. However, there are weak spots with various subsets of pulmonary embolism and prosthetic valves. For instance, prosthetic valves, echocardiography can't distinguish between thrombus and pannus. Pannus is a fibrous tissue growth that affects prosthetic valves. It looks the same as thrombus. in an echocardiogram. The problem is they're treated very, very differently. And the only way you can distinguish them right now is with surgical intervention. We can distinguish between the two of them with FibroScent. So these are the things we would initially be up against in terms of competition. But again, we feel like once we establish the value of this, we have the chance to transition to being the tool of first choice. Our intellectual property position, we're in good shape. This was developed in the laboratory of one of our founders, Dr. Lanza at the Washington School of Medicine. Washington University owns the rights, and Capella Imaging has the exclusive license to develop and market it. We have made some structural improvements to the original molecule in the course of developing a scalable GMP manufacturing process that we will be filing a composition of matter patent on to extend our protection lifetime. But we're in good position with protecting our intellectual property. At this point, we're looking for a $500,000 investment in the first quarter of next year. We're going to use this to support the exploratory IND submission and phase 1 clinical trial. Exploratory IND, I want to kind of stress that if you're not familiar with it, it's a very flexible tool. We can, once we have an exploratory IND, and our initial one will be for the LVAD indication, that permits us to get, our phase 0 data with healthy volunteers, and we will follow that with a group of 20 patients, 10 that are positive for left atrial appendage blockage and 10 that are LVAD patients. And, after success with that, we will transition to a full-blown IND in phase 2. But the beauty of this is we can simply modify the protocol for subsequent indications. We will update the protocol for doing prosthetic valves, and we already have partners here at the medical school that are poised to do that work. So it's a very useful tool, the exploratory IND, for doing this kind of work. This is the FibroScent, the Capella team. Is one of my business advisors told me it's one good guy and a bunch of schmucks. On any given day, I can't decide who the good guy is, but let me spend more time on our advisors. This is a world-class group of guys. Dr. Siegel and Sally Schwartz are just household names with the FDA. Sally has actually written the guidance documents for exploratory INDs. Dr. Ewald is the head of the Heart Failure and Mechanical Circulatory Support Program at WashU, and is the gentleman that actually deals with all the LVADs. Dr. Zaharias is the head of the Valvular Heart Disease Center and is the gentleman that will drive the prosthetic valve program. And Dr. Schindler is an expert in PET and SPECT imaging, and this is a SPECT imaging. SPECT is the technique we use for imaging. I kind of want to stress that. SPECT is, we're using technetium. Technetium, I think there's like 120,000 medical images a day in the US that are acquired using technetium. Nuclear imaging is kind of sexy right now, and there's a lot of stuff out there that uses very high energy, very exotic isotopes that are logistically very challenging. Very short half-lives, have to be, you have to have an accelerator nearby. Technetium is a catalog item with Cardinal Health, and the SPECT cameras, they're about 1/10 of the cost of a PET camera, and they're very common. So we think SPECT imaging actually gives us quite an advantage over PET imaging. And I think I will stop there. Thanks.
Thanks, James. That's been fantastic. Thanks so much for that great walkthrough. I love the word de-risked. That's one of my favorite words. That's like free beer in my head. De-risked. That's just fantastic. And we're going to get to that in a minute. But first, I'm going to talk a little bit about the science aspect and some IP. So talk to me a little bit about why your molecule is able to differentiate between the fibrin that's in a clot versus those in other related types of fibrous buildups that we might occur, like around valves and whatnot. Why is it so selective?
Well, that's actually an interesting point. So you're assuming that it can, and we so there's actually been a fair amount of interest, for instance, in people that look at multiple sclerosis, because there's fibrin buildup. around, the demyelinated spots associated with multiple sclerosis. And, we've got a bandwidth problem, truthfully. There's some folks doing some outstanding work in San Francisco that, we're still in discussions with about providing material. But, this is one of the things I've really enjoyed about this as a chemist is, I've got a long history in big pharma where we tend to make molecules and then we go and market them to the masses.
Right.
Whereas in this case, this came up this all started with a conversation between Drs. Ewald and Drs. Lanza. And they said, look, we need something to help these LVAD patients. And where these other indications keep coming from is colleagues at the Washington University Medical School that hear, they get word of what we're doing and they come and say, hey, look, I think you can help us here. And so it seems like every 3 months, we're adding something else that we would like to explore. And so to answer your question is it's really not necessarily that specific. It's not that it doesn't recognize fibrin elsewhere. We think we can pick up those deposits. We haven't done the studies yet, but there's a lot of interest in actually doing that, especially, multiple sclerosis, Alzheimer's disease is one. Yeah.
Okay, so if that's the case, you have technetium, you have a fibrin detector. One of my favorite questions that I like to ask my companies is, what makes you so effing special? Like, why hasn't somebody else done this before? Because it feels like what you've done is link two molecules that are already in use together and magic happens. What's going on here?
Well, I mean, the magic is actually the targeting vector that recognizes fibrin. And again, it's not just acute fibrin, because there have been things around that do that for a while. We do it with a great deal of specificity, a great deal of avidity, and again, we're agnostic to whether it's fresh or old.
Got it. And like having 2 images a year, like I totally understand first use case LVAD. I mean, here you've got a situation where they're already compromised in many ways, both from anti-clotting and other drugs going on, and you've got a pump which can, in many circumstances, create additional clotting risks, right? So
Yep.
Perfect use case. But in that situation, why would we believe that 2 images a year is sufficient, or that they would need as many as 2 a year? Like, meaning, like, why 2 a year? Why is that a sweet spot in terms? Remember, we're building your valuation matrix off of that 2 a year kind of total.
Sure. And so that's a good question. And until you actually do the work, we're not sure. I mean, I was pressed on this by Abbott not too long ago. They showed me an image, and is this good or is this bad? And I'm like, well, hell, that's a good question. We got to get out in the field and start doing the work. So right now, I mean, our feeling is with again, let's focus on the HeartMate device. About a third of those patients will end up being hospitalized to treat gastrointestinal bleeding. And so our idea, the way
just as a reminder, gastrointestinal bleeding as a result of excessive use of antithrombosis.
The anticoagulation therapy, that's right, because these patients are treated with very high doses because there's no way to assess how well it's working. Mm-hmm. And so our feeling is you get implanted with an LVAD, once you're stable, you get image. Okay, this is our baseline picture of this device. And you're placed on anticoagulation therapy. You come back 6 months later, you get an image again, you're clean. But maybe you've had a couple of gastrointestinal bleeding events, nothing too serious, but regardless, your pump is clean. At this point, you can titrate the anticoagulant down. You reduce it a bit, you come back 6 months, maybe it's 3 months. I don't know. It's the kind of thing we have to actually get in the clinic and do the work to find out what the optimal frequency is. We figure at least 2 images a year. But eventually you want to titrate it down, ideally, until you get to a point where either A, perhaps you start seeing some thrombus, or B, you just get to a point where there's absolutely, no indication of gastrointestinal bleeding, and you've got the best of both worlds there.
Right. So there's been a lot of we need to find out, we need to see, which makes me think of two very important questions. One is more of an observation that we really need to get some doctors involved in a focus group. And I know that was something that you really wanted to get done as well. So doctors on the line here, or any of your cardiologist friends, or anybody that watches this video in the future, if you want to get involved with helping James with an evaluation on this company, it's a paid evaluation. We're establishing how much that's going to be. It's probably not a lot. It's probably going to be more of a focus group, but it will be a paid evaluation. You will get involved in the evaluation process where the pay can go up to as much as $1,000 of equity in these companies. But for now, we just want you to know that we want your opinion, and James desperately wants your input in terms of use cases, adoption rates, adoption hurdles, whether you find the value in this product or not. I certainly do. I think this is very interesting. We have another company coming up that's also in LVAD. I'm sure they're going to see that as well. But let me ask you the kind of the harder question that I wanted to ask on that front. There's been a lot of unknown that you've said. So how is this de-risked? You open with my favorite word in the human language. You've been de-risked investment. How are you de-risked if you still have this many unknowns in terms of use case and other things?
Well, so my feeling is when I say unknown, it's not really a question of the utility, or is this going to work? It's a question of optimizing how's the best way to use this tool. And realistically, it probably differs from indication. With LVADs, I'm sure we will land on a number of images per year that are deemed adequate, right, for evaluating the anticoagulant therapy and the associated risk to the patient. For prosthetic valves, as an example, which we think is the follow-on, is the next indication, that's likely to vary depending on the type of the valve, the location, the time from placement. So, these are details that kind of define whether it's going to be merely big or really big, but it's clearly going to have value. It's just going to work. I mean, that's what I love is again, if you look at this preclinical model, the damn thing works. And so, the utility probably, how it's optimally used will vary depending on the indication. But there's really no question. Again, all these indications came, as an example, Dr. Zaharias came to us and said, "Hey, James,
I'm sorry I have to cut you off, and even after I just asked that question, I'm so interested in the answer. But I want to be respectful for our third presenter's time this evening.
Absolutely.
I think the right answer, and I'm just going to put it back into my own language. You've de-risked the use case, but there's a lot of unknowns in terms of how many different ways this could be used. So you've got a bit of an embarrassment of riches in your unknowns, is what you're kind of telling me, right?
Some problems are good.
We don't know. Yeah, we don't know if 2 is enough. Maybe you'll need to get scanned 4 times a year. That would just double your potential total addressable market. We don't know how many other different products it could be used. We know it's useful for LVADs, but it could be used for all of these other indications as well, which again could expand your total addressable market. I like that kind of unknown. That's a good risk. Upside risk is always a good risk to have. So there is that, right?
Very good.
Excellent. Hall, I'm sorry for eating up the time with all my questions, but please, my apologies to Windmill as well.
No, great. Thanks, James, for joining us. We'll see you soon in the breakout rooms. Next is Dr. Richard Smalling and Sam Lang of Windmill. If we have Dr. Smalling and Sam up here, we'll have you guys kick off with your presentation. I think we may be having shorter breakout rooms, but we'll maximize our presentation time. With that, go ahead, guys, kick off.
Great. So I'm Richard Smalling. I'm an interventional cardiologist and have been in the LVAD exploration science for over 40 years. We've developed a pump which we'll describe to you in a minute. And let's go to the next slide. You've already heard about the need for left ventricular assistance in patients with end-stage heart failure. And about 6.5 million people in the United States suffer with that. About, to get down to the sickest of them, about 50,000 have end-stage heart failure, and they're not going to be able to survive without a heart transplant or an LVAD. Only about 4,100 get a heart transplant because there's just not enough hearts to go around, and about 4,600 get an LVAD for a lot of reasons, which we'll go into in some detail. That means about 40,000 are condemned to palliative care, which means they say, I'm just not going to mess with that complicated technology. I don't want to carry a big, heavy pump around with me for the rest of my life. Just let me go. Next slide. So the mission obviously is to extend the life and improve the quality of life for patients with end-stage heart failure. And we have developed a revolutionary pump that should solve most, if not all, the problems associated with LVADs at the current time. We talked about heart transplant already and the LVADs, and as you can see by this slide, the current devices require 2 batteries, a controller, transcutaneous cable, and so forth. It's very, very user unfriendly. Next slide. So all of the continuous flow pumps basically have very high shear rates. They run at about 3,000 to 8,000 RPM. They have very small gaps, and so as a result, they pretty much trash the blood. They lyse red cells and cause significant hemolysis. They deplete von Willebrand factor and produce von Willebrand factor fragments, which in turn induce AV malformations in the gut, which leads to almost a 40% incidence of GI bleeding. They activate platelets that cause stroke, and they also damage white cells, which result in infection. And you can see from the Intermax 5-year data, the stroke, thrombotic complications, GI bleeding, and infections are major issues, and the rehospitalization rate is just unacceptable at 93%. Next slide. So up in the left-hand corner, you can see a cartoon of our pump. It consists of 2 pistons that are independently controlled. One serves as a virtual valve between the inflow cannula and the outflow cannula, and they switch places as they make the circuit. The pistons are precisely controlled, continuously monitor transpump pressure, and we have an existing ECG lead that is implanted usually on the surface of the right or left ventricle, which then allows the pump to trigger its pumping function with the ECG of the heart. So the pump is designed to pump in early diastole, which allows the heart to pump on its own, inject blood through the aortic valve. So with our pump, in contrast to all the other pumps, the aortic valve opens with every beat, so we don't have a problem with ascending aortic thrombosis. It also eliminates the risk of right heart failure due to septal shift. Continuous flow pumps overpump the left ventricle, leading to sphericalization of the right ventricle, which causes it to fail. We've done a number of bench tests and blood compatibility tests. Carlo Bartoli, one of the thought leaders in LVADs and one of the world experts in blood traumas, analyzed our stuff in addition to us. And you can just see that the von Willebrand factor depletion with increasing flow rates with the HeartMate 3 pump basically completely obliterates the von Willebrand factor while we barely touch it. So there's no question that we will not activate platelets, will not activate white cells, and the risk of thrombosis in addition is much lower. You can see on the upper right the small platform that we have developed and a small battery compared to the current HeartMate 3. Next slide. The engineers, I asked the engineers to test the pumping raisins to show it doesn't hurt big objects, and they followed that with pumping a fish. And so as you can see, there's no blood trauma in the fish, and it's a very, very mechanically gentle pump. Next slide. I guess it's not running quite well, but if you can see it here, comes the fish. Next slide. So we have several pump iterations. The generation 1, which is the one we showed previously, has been tested in over 50 animals. We'll talk a little bit more about that. We also have a version of a totally implantable system that's has a patent that's been submitted, and we have also developed and tested a smaller version of the Torvad designed for children and women. Only 20% of eligible women get pumps that need LVADs, and that's largely due to the fact that they're smaller and the current pumps just don't fit in them. Next slide. So we've talked about our 3 generations. So we have 5 issued patents, 1 pending, 5 in process, a landscape analysis that Sam will talk about more in detail. We've gone through, as we mentioned, more than 50 animal implants and have determined on the bench that the pumps have lasted at least 24 months with no failures and no controller failures. We presented nationally and internationally at almost 30 to 40 meetings, and we have 10 publications about the pump. We have engaged a regulatory company called MICRA, comprised of largely folks from the FDA who were in the actually ran the circulatory assist device approval program, and we have contracted with Valenium, which does manufacturing of medical devices here in Houston, and also is very acquainted with LVAD manufacturing. So the Valenium will be making the commercial versions of our pump. I will tell you that most of our funding has been through NIH grants, as well as from individual contracts with various companies, and so I think that will lead to an attractive funding situation for potential investors. Next slide. These are just comments from key opinion leaders that have been independently queried by one of our companies that advised us on the development of the device. And uniformly, all of them said, if we had this pump, this is the only one we'd use, and we'd use it in a lot more people than we're currently putting it in. There's been absolutely no negative comments regarding the pump. Next slide. So
I'll
Sam,
go ahead. Yeah, I'll go ahead and pick it up from here. Yeah, Richard. So the current market today, it is a reimbursed $1.1 billion market, with over half the market, about 600 million here in the US. If you actually look at the number of patients treated, as Richard shared in the opening slide, only 9% of the patients are getting LVADs. And so the actual total available market in the US alone is 6.4 billion. With the launch of Torvad, we firmly believe, based upon the voice of customer research from several KOLs, this could actually replace HeartMate 3 as their first-line therapy. But more importantly, as Richard just shared, it would expand the use of LVADs, primarily due to reduced adverse events. And so post-launch, within a year, and a modest penetration from going from 9 to 12%, the TAM actually can continue to grow to well over 1.1 billion in the US market alone. The company is currently raising $7 million to hire an experienced CEO, as Dr. Schmalling is still a practicing full-time cardiologist, and it is to execute these 2 agreements with MICRA and Valenium, who are extremely qualified suppliers that have LVAD experience. Together with the Windmill team, they will plan to finalize the product design, the pump control, and the peripherals, and develop the EFS protocol, which is for first in human. Should note that the potential exists on several dimensions, which I can outline later on during the Q&A sessions, but all of the exits that have occurred in the market, have been in the billions of dollars. Multiple companies do call on the strategic call point and have expressed interest in the company post first in human in getting involved in more detail with the company. Besides the deep experience within the windmill leadership, I think what's also important is if you were to look at the lower level, these are the thought leaders that are in the mechanical circulatory space. So just in summary, like to turn it over for Q&A. I think we've talked through the fact that there's a large unmet need. The TORVAD, which simulates the human heart, pulsatile flow, rate responsive, and also has the ability to be delivered via a thoracotomy as opposed to a sternotomy, will certainly take share from the current player, Abbott, which is a single player market with just Abbott alone, as well as expand the market for patients worldwide. Thank you.
So you mentioned two things there at the end that I'd like to talk a little bit from a science and medical standpoint.
Sure.
We're not going to have to crack the sternum to put one of these in?
No. All of the implants have been done through a lateral thoracotomy. I'm an interventional cardiologist, but I've implanted most of them. So it's not that difficult. Basically, we'd use a pre-peritoneal pocket, put the pump in, anastomose the outflow graft to the descending aorta, and then put an apical sewing ring on the apex of the heart that has a magnetic coupler. While the heart's beating, actually gently lift up the heart, put an 11 blade through the apex, put in a coring tool, core the apex, put your thumb over the cannula connector, and then pop in the inflow cannula into the heart, and it automatically connects, and then you go on pump. We've never had to put any of the animals on bypass to implant the pump. So it's quite easy, actually.
Wow, so you're actually installing with the heart still pumping away. You haven't even had to stop it and put it on the bypass.
Exactly right. Mm-hmm.
Remarkable. And then you mentioned another thing as well that I thought was phenomenal, which was the fact that you can have pulse responsivity.
Yes.
So are we fully mimicking the actual natural heart rhythm? And how do you do that? How do you set it up? How do you sense it and react?
So it has the ECG lead that goes into the controller, into the pump actually, which then is processed by the controller. So it looks and approximately
you're looking for the response from the heart itself, using that as the stimulus to
right. So right about 80 milliseconds after the QRS, the pump will actually pump, and that the aortic valve at that point will be closed. And then as soon as the pump pumps 30 cc's, it will stop. And then the heart itself contracts, the aortic valve opens, and there's normal flow. But in addition, it has built-in intelligence so that if the heart rate is too slow, and the pressure goes down, it will switch to asynchronous mode and pump at a rate sufficient to maintain a mean pressure of 70. Conversely, if the patient goes into supraventricular tachycardia, it will go into Winkie-Bach mode and pump every other beat or whatever, and at the same time monitoring mean pressure. So if the pressure is sufficient, then it may not pump, but once every other or 3 times beat. So it's highly sophisticated.
So basically, you're allowing the natural rhythm of the heart to stimulate the aortal assist, and then you're creating barriers in order to keep it from staying within range.
Right, exactly.
Okay, phenomenal. And I mean, sounds to me like both of those are extraordinarily powerful arguments for increased use case, to the extent of almost like replacing like any use of artificial hearts or other more complicated devices, certainly.
That's correct. Well, the other interesting thing, it also transmit via Bluetooth every 8 seconds, heart rate, rhythm, blood pressure, motor currents, and a number of other factors to the monitoring center And so to give you an example, after our first animal implant, we got back to the hotel to a restaurant, and the engineer's phone started ringing. The controller was calling him saying there was a major controller issue. So he had to go running back, fix the problem, and then got the pump back online. So that's all. Nobody has anything like that. So you can look at trends and so forth as you wish. So it's very, very, very helpful.
Fascinating. And existing, so I don't see how you guys get to market before you're acquired, right? I assume, Sam, that you're expecting that a competitor takes you out?
It would be. So I don't know if people can see the slides as far as how we've laid out the financing and the key milestones that are here, but the $7 million tranche, tranched in two tranches, 3.5 and 3.5, will take the company essentially into 2026, at which case there's another about 13 million that will be needed in total here to complete the first in human trials.
Sorry, before you get into that, I do want to talk about this timeline in a little more detail in a moment, but I want to stay on that for a second, because I'm worried about a smaller company's ability to go to in-human, right? In your other competitors, the other companies that were taken out, were they generally acquired after in-human or before in-human?
They were acquired post-in-human.
Post-in-human. So you're pretty convinced you're going to have to go into human, even though the methodology and the technology is fully stacked. I mean, there's nothing special here in terms of conceptually, right?
Yes, Michael. I think, and also, you know, for medtech investors, they would, at this point in time, the bar is kind of raised to people want to see in human data, improve the value proposition.
All right.
Yep.
I mean, there are some devices that are external that I think are dangerous beyond belief, and there are some internal devices that feels to me like it should go through a de novo protocol. And I know you're dealing with a heart replacement product; you can't go into de novo and just get it easy. But this certainly feels like it. You're moving the blood. It's less, you're creating less clots. How is this possibly more dangerous than the existing product? Right.
Right.
All right, so there is a little bit of an extended timeline that we're going to have to wait on you guys. So investors would have to be somewhat patient.
Yes, and I mean, just for full transparency, as I mentioned, we're raising 7 million. Another 13 million is there to complete first in human, and then what you can see at the very bottom, you know, post first in human, the company will be at several inflection points where we can continue to go through VC strategic financing, where any of the strategics want to be part of the company. There's certainly an IPO market that could be rebound by the 2027 timeframe. There could be an exit, but also,
dear God, I hope it rebounds by 2027.
Yes, we hope so. Absolutely. And then the very last one is this is a very interesting investment vehicle. I'm a operating partner at a company at a PE firm called Revival Healthcare Capital, and basically we have a vehicle which is a structured exit called the build to buy, where post first in human, a strategic would say, look, we know we want Torvad, but yet we're not ready to acquire it. Revival will put in the capital, and essentially at the time we would execute a structured exit at that point in time, in which case Revival would put in the capital to run the US pivotal, and then post approvals, assuming it's successful, a successful trial, the strategic would basically execute a call option to fully acquire the company. But it is at this point in time, the difference is it's not like a rights. At this point in time, the difference is here, there's actually a structured deal. So everybody knows what the exit price, what the strike price, what everything what everybody's playing for.
And what's your valuation right now that you're looking to come in at?
Yes. Yeah, the current valuation that we have, we've raised about 14 to 15 million, most of it non-dilutive financing, and the valuation is at 19 million. So very, very, very modest step up. Given
so
all the work,
we're having to wait until 27.
Yeah,
we'd have to wait till 27 or so to know the exit structure, but we're still looking at.
Yes.
I mean, not many device companies that you're looking at 100x return potentially here, and that's what we're talking about, right?
Yeah, it is a very large return because it's a large unmet need.
Right. I mean, obviously, you'd have to stay along the line and not get diluted out, but you're still talking about a very, very, very nice return. Excellent. Now, would you like to talk to some more doctors that are doing this product, or like, you've got a stacked board, right? You've got stacked experts already involved.
Yeah.
Are there any doctors you would like to talk to more?
I mean, go ahead. I've got some opinions, but go ahead, Richard.
Yeah, so basically every doctor or every transplant physician, surgeon, heart failure specialist has seen this, says, when can we have it? I want it tomorrow.
Right.
And then the pediatric cardiologists are just crying because they have nothing. And
Right.
so there's not been anybody that hasn't been willing to step up to the plate. I have a whole lot of friends. We've been doing this for 20 years. So I've got a whole lot of friends in the space. I've helped develop, in fact, I helped develop the hemopump, which was the predecessor to Impella. So
I have to be honest, Richard, I was a little bit, sorry, Dr. Schmanske, I was a little bit surprised you guys weren't talking about breakthrough use case for the pediatric.
Well, we actually, we are talking about breakthrough for the adult product. We certainly would look at breakthrough for the pediatric product. We're also exploring other ways of getting to first in human OUS, which should accelerate the pathways considerably, but we're very in the early stages of that effort.
What about a CMARC in order to go to market faster in Europe instead?
I mean, I can comment on that. I mean, with the recent MDR, which is the new, approval pathway, it is just as rigorous as the US. So going US is still, the first and primary focus market.
Okay. So no longer the case that you can get these in use in some clinics in Switzerland and collect the data. Right?
No.
That was a nice period of time. That's a shame that's over. Well, fantastic, gentlemen. This has been wonderful. And again, a reminder, doctors, that if you are interested in investing or learning more about these companies, all of them have opportunities for additional warrant coverage if you are a specialist in their space. They want you. Your specialty is important. Your feedback is important. Your buy-in is important. All these guys, they know that the best way to get to market is get the buy-in from the specialists up front. Use that to your advantage. Invest in these companies now. Get the extra equity and help them make a success. And with that, I'm going to hand it all back to Hall. Gentlemen, thank you so much for joining us tonight. I think you've got a remarkable product there, and I'd love to hear more about you guys in the future.
Thank you very much.
Thank you.
Great. Thanks, Sam. Thanks, Richard. Michael, thanks for hosting today. Appreciate the great Q&A sessions with each of the companies that we've had here today. So I guess with that, we're wrapping up for today. Any closing thoughts, Michael?
No, I think you've done a remarkable job summing up everything I had to say. I mean, I just want to lay down. It's really easy to contact us. Oh, one other thing. Both AngelMD, Hall, and MD Capital will be running newsletters on a regular basis with additional opportunities for investment, additional opportunities to learn more about companies, including these three studs. So make sure that you contact us if you want to be on any of those mailing lists as well. That's all I have to say. Thank you.
Great. All right. Well, great. Well, with that, we're going to close it out for tonight. Thanks so much, guys, for a great session, and looking forward to seeing you next time. Have a good one.
Thank you.